Okay, thanks everybody for coming to our talk today. We're kind of getting to the end of the quarter, end of the year, and also the time of year when it's just absolutely sort of stupidly beautiful outside. So thanks everybody for being here in two or three dimensions. We're excited to have Jeff here today, Jeff Leek. And Jeff has a lot of accolades that I can talk about. But but instead, I think I'll just tell you one thing about Jeff, which I appreciate. I've had the privilege to know Jeff for a while now, and Jeff's one of those collaborators or people that you walk into their office and they're excited to talk to you about things that they claim to know nothing about, but certainly will explain to you patiently over and over again something that you know nothing about. So I don't think I could explain to you what Jeff's scientific interests are, which are deep and profound, and his contributions have been substantial beyond maybe like a first or second grade level. But nonetheless, I've had a ton of extremely rich conversations with Jeff where I've learned a ton, or I've learned a lot of things, and I think that's a very enriching thing to have in a colleague. So I will set that up as I don't know what's about to happen, but I've heard it's going to be interesting.
Yeah.
So thanks, Jeff, for coming.
Yeah. Thank you for having me, Tyler. I appreciate it. That was first a lovely introduction. It's nice to see you all. Hopefully everybody online can hear us okay. I think in light of a couple of things. One, Tyler and I have far-ranging interests and talk to each other about lots of stuff, some of which is really statistical and some of which is not. In light of that, and the fact that I didn't know what the audience was going to be like, and the fact that it's Friday afternoon and I didn't know if anybody was going to be here, I decided to just talk about what I'm interested in right now. And so I'm going to tell you about some things that are related to bigger projects we're doing at the Hutch, which I hope will be, and nationally, which I hope will be interesting to some people to collaborate with us on. And then I'm going to talk at the end a little bit about a statistical idea that Tyler and myself and a large group of our students and postdocs have been focused on for the last couple of years now. And so there'll be a lot of stats at the end, but at the beginning, it's probably not too equations and stats heavy. So and it's going to be a little bit weird. So I'm excited to be here. Thank you for having me, even though it's Friday. And thank you for everybody to coming here at this time of day on a beautiful day. There's this great quote, and this is kind of an alternative title to my talk. So this actually came from Tian Zhang, who I was giving a talk with at ENAR, and she had her title of her talk was "Statistics is what she didn't have the bio, what statisticians do." And she was talking about how the statistics as a discipline has really expanded beyond its sort of roots to be doing lots of different things, especially in this sort of AI age. And so you're going to hear today about lots of things, many of which aren't like statistical algorithms or statistical theorems, but are things that a biostatistician is doing. So the sort of outline of the talk is, I'm going to talk about, I know the title is an AI engine and like, what even is that? So I'm going to talk a little bit about what that is. Then I'm going to talk about how we're building those at the Fred Hutch and scaling them nationally. Then I'm going to talk a little bit about what you can do with one once you have it. And then finally, the stats part is, what do we do after we machine learned everything? So I'm going to talk a little bit about this throughout the talk here today, and hopefully there'll be some interesting stories for you to take home. So it's going to be weird, and we'll see how it goes. So one thing that's important for part of this talk is this is a thing that if you go to biomedical talks, they always have these conflict of interest declarations, but I have some. One of which is going to be relevant for the talk today. I'm going to talk a little bit about some of the work that's being done at this for-profit company that spun out of my lab and another lab at the Fred Hutch. And so for that part, you can assume it's an ad for a for-profit company and choose to believe it or not as much as you choose to believe it or not. So I'm going to talk a little bit about these building an AI engine and what does that mean. But the first thing I think is useful sometimes is to ground people, like what is AI? Because you hear it a lot in the news. There are many, many different definitions of AI. The one that I like best is really simple. Anything that has these three parts is an artificial intelligence. It has to have some data that is being used to train an algorithm. It has to have an algorithm being trained. And then it has to have an interface to that algorithm that humans can interact with. And so in this case, I'm showing you the ChatGPT. They trained on all the text on the internet. They downloaded it and chopped it up into little bitty pieces and stored it all. They used the transformer algorithm, which is is all you need, that sort of decides how you can fit models to this kind of data very rapidly and scalably. And then the interface we all probably know is something like ChatGPT, where you're interacting with a model. You could also do it for like self-driving cars. It's like videos that you collect from the car, algorithms quite similar. and then it tells your car to turn the steering wheel left or right. So in any case, these three things kind of define an AI for me. And why is AI in the news? There's kind of two reasons. One is there's these two revolutions happening at the same time, which is kind of head spinning if you work in the area I work in, which is at the intersection of computation and biology. On the one hand, you can rent more and more computers on the internet, and they're getting cheaper and cheaper to rent. And so you can rent like enormous amounts of computers. You can go on the internet and rent 10,000 computers from Amazon tomorrow with a credit card, which is kind of nuts. You couldn't do that a long time ago. And that allows you to train really big algorithms. And on the other hand, data is getting cheaper to collect than ever was. So when I started my career and when I started working in this area was in 2003 when I was a grad student here at UW. And the cost of sequencing a genome was like somewhere around $100 million. And now it's like less than $500. And so that precipitous drop in the cost of collecting this data means that people collect a lot of it because it's cheap. So you could replace this is genomic data. You could replace it with imaging, text, video, anything else. Everything's getting cheaper to collect. So we're just getting inundated with data and we can do stuff with it with computers. So you have this big revolution. So it feels like this almost all the time in the places where I work. Like everybody's like trying to freak out and figure out how do you use these things and do interesting, new, cool stuff. But AI is something that's come back over and over in my field, probably in other people's fields. And so when people talk about AI in biomedical research, they say, well, haven't we seen this before? There was this big push just a few years ago. This is 2015, about 10 years ago, where there was something called IBM Watson, which was a thing that IBM claimed was an AI that was going to come and like cure all of the cancers in the world. And ultimately, it didn't end up working out. Like they didn't do very well. But people were really worried for a while that they were going to shut down all biomedical research because Watson was just going to solve it all. And it didn't really work out that way, largely because a lot of the work that Watson was doing turned out to be humans in the background doing a bunch of work and then feeding that work into an algorithm that would then make very small scale predictions. And if those humans weren't around, then the algorithms didn't work. And so it ultimately wasn't a big impact on the field. And so the robots weren't quite ready to take over the jobs just yet. And if you don't know Simone Gertz, this is probably the best part of my talk. You should go watch her YouTube channel, which is unbelievable. She makes these crazy robots that are really fun to watch. So why am I still excited about AI? Why am I talking about AI given those things that have happened in the past? And I want to talk a little bit about a story that is about somebody else's story for a minute, but is what inspired me to come to the Fred Hutch and not just be a professor anymore, but also be a chief data officer. So this is a person named Suchi Saria. She's a really accomplished expert in AI and healthcare. She and I started on the faculty around the same time at Johns Hopkins. She published this paper in 2015 that was wildly important, where she developed a way to take the ambient measurements that they would collect about patients in health systems. So like think the little like blood pressure cuff they put on you, think the like blood measurements they're taking, think the temperature in the room, pH from your blood, things like that. And she could predict early up to 6 hours, up to 8 hours early if you were going to have a septic shock, which is a shock is essentially you get blood poisoning from an infection. So she could predict that early. And it was a huge hit. Everybody was really excited about it. And then for like a long time, nobody heard anything from Suchi about this, which was very shocking because this was such a big paper. Everybody was talking about it and like nothing was coming about this. She was publishing other things, but nothing about this for a long time. And then in 2022, she published what I consider probably like the greatest tour de force in the area of translational AI in medicine that's ever been published. And so she had engineered her warning system into the Johns Hopkins healthcare system. She had studied the way that physicians interacted with the alerts that came from that health system. And then she'd run a prospective randomized study to prove that the alerts from her score would reduce morbidity and mortality with like a causal analysis. And so this dropped like, I mean, in the field, everybody was talking about this. It was crazy. It was so amazing. And I'll tell you a little bit more about it in a minute. But the main thing that was relevant to me and my decision to come here is the dates, 2015, 2022. So it took 7 years for her to do this work. And why did it take 7 years? Because all of the underlying stuff that you need to do to enable these kinds of studies is horribly slow, bureaucratic, technically difficult, et cetera. And so people could only do these only if you had the determination of Suchi and you had 7 years on your hands, could you do a project like this. And So this idea of an AI engine became what kind of inspired me. And I didn't have a name for it. I actually borrowed that name from this person, Andre Karpathy, who used to be at Stanford. I tried to recruit him to be a faculty member at JHU when I was there. And he said, I have a little something else lined up. Turned out to be the director of AI at Tesla. Worked out okay for him. And he had this idea of an AI engine where you have the cars, the Tesla cars. You collect some data from the cars. That's the videos. You do some kind of labeling and cleaning. You build models, you put them back in the cars, you collect more images, and you repeat over and over again. And so most of the time when we're building machine learning models or AI models, you don't do this multiple times. Like if you work in machine learning in our field, usually you collect one big data set, you build a model, you publish a paper, done. And his argument was that you need to be able to cycle through this more frequently. And so when I came here about 3 years ago, the ambition was to try to build an AI engine for the Fred Hutch that would let us do this cycle more rapidly, kind of like Suchi had done, but not once every 7 years, but rather maybe once every month or once every week, or how fast could we do it? So to do this, we have to build things for each piece of this. So first for collecting the data, and there's a whole bunch of components to how do you actually collect and use health data that make that not just a matter of like taking the data from electronic health record and dumping it somewhere, which is like very fast. You can do that in a day. But it's challenging because you need to be able to set up the systems that are secure. You need to make sure that the data are modeled in ways that people can understand it. There's lots of regulations, ethical issues, and then you have to have a data governance framework to say who gets access to which data and all that kind of stuff. So we had to build all of that to sort of enable people to use the data. And this turned out to be like one of the biggest needs at the Fred Hutch. Turns out it's a big need at many cancer centers. This is data from a survey that we did when I first started as chief data officer. We went out and interviewed tons of people all across the Fred Hutch, everybody from faculty to students to trainee to postdocs to technicians to administrators to lawyers. And we asked them, what are the things you need most help with data? And that quickly at the very top, they said, we need to be able to like get access to data. We need to know who has access to it, and we need to be able to analyze it on a secure infrastructure. So we spent a long time building things like this to help people use the biomedical data to make discoveries. And what we had to do was build what's called, in the term of art, is a data lake. We take a bunch of data from UW Medicine, from Fred Hutch Cancer Center, from pathology department, from external companies. We put it all into a big system that's on Amazon so that people can access it and interact with it. And then we build a governance system around that so only the right people get access to the right data. This is in one slide, 3 years worth of labor. And then we enable people to do that by connecting all the tools that they like, some of which are like cBioportal, some of which are Databricks. If you are in this area, you know what these tools are, Red Hat, things like that, so that people can interact with the data. So the goal is to build what we call a pit of success. So this is a term of art that comes from Hadley Wickham, who, if you know, he's a statistician who writes software. Hadley has this term of pit of success, which means the easy thing to do should be the right thing to do. And so it turns out making mountains of success is quite easy to do. You tell people just do these 45 processes, do them in the perfect order, don't miss one form, and you can do the thing. Like, that's quite easy to do. Setting it up so people fill out one form and they get access to the data they want in 24 hours is digging a pit of success, and that's much, much harder to do. So we've spent a lot of time building pits of success. The next part is like, okay, we have that data. What do we do with it? What discoveries do we need to make? And the thing that's really interesting about this is most of the innovation in this area is around what data is available to you. And so this is from Suchi's paper. They collected very basic data. Like, these are measurements that are taken routinely in the clinic. And using that was able to build models that predicted this septic shock. And so the interesting thing is much of the ambient data that she was collecting for this study, it was being collected by machines, but never aggregated anywhere for anybody to be able to use it and model it. And so we spent a lot of time figuring out how to aggregate all these different data sources just to make them available to interested scientists who can get involved and try to figure out new machine learning methods to build. Then we have to engineer that into something. So, okay, we've made a discovery, we've made a machine learning model. This is the part that took Suchi a long time. We have to set it up so that we can plug it back into the health system. And so that's what we're working on now at the Fred Hutch, is ways to take machine learning models that we develop and deploy them back into the health system. One of the things that's really interesting about that is there's a huge component of what's called human-computer interaction research around that. So the big challenge is you can give people an alert, like you say, your patient's going to have septic shock. And most of the physicians will ignore that alert because they're getting like 4,000 alerts. They're like, might get septic shock, might have this, might have that other thing. So they just close, 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 close. It's like, I don't know if you remember the old browsers where all the spam windows would open up. It's like that. And so she did this study to figure out when would people use these alerts, what are the best way to use these alerts. And so found some interesting findings in that study of how humans interact with the technology, and discovered that, like, obviously if it's a high red alert, they paid more attention. But also interesting things like if it was the beginning of a shift, the alerts worked, and if the alerts kind of didn't work. And also like certain physicians would use it and certain physicians just wouldn't use it. And so like there are all these factors that go into, even if the model is accurate, even if it does well, even if it's going to help, you still have to understand how a human interacts with the computer. So we have a little HCI group at the Fred Hutch who's like doing this part of it, trying to figure out when we build things, how do the humans actually interact with them? And then finally, we have to validate it. You know, in self-driving cars, you can probably slap a GoPro on the top of your Prius and call it a self-driving car in the state of Arizona. The regulations are pretty light. But for human health, we have to be a little bit more careful. And so we have to do randomized trials, prospective studies to validate that the things that we're showing people actually have impact. So she did that in this study, and that's sort of what we're setting up now, is to be able to run these randomized trials in what's called a pragmatic trial, where you show the information in a randomized way. Some people get the alert, some people don't. And then you say, if you got the alert, do you see differences in morbidity and mortality? Which she showed for their alert that they had these quite significant reductions in morbidity and mortality for what was a pretty like straightforward prediction that was helping them. And so Suchi ended up starting a company off of this. It's now being used at hospitals all over the country. It's kind of amazing. But our goal is to build an engine so at the Fred Hutch and people who collaborate with the Fred Hutch, we can do this many, many times over, not just once or twice. And one of the things that was so important about this work was really understanding in a deep way the trust that needs to be built between machine learning systems and humans, which turns out, like in this time of when AI is being used all over the place, we still don't really understand when people trust it, when they don't, when it makes sense to use it, when they don't. And so this was a really key study in figuring out how physicians would interact with these sort of machine learning models. It was a very cool study. So we've been building all of that at the Fred Hutch, and then we've also been building software at a small scale that helps people build these machine learning models. So one of my postdocs, Steve Salerno, has been working on this software called Scorcher, and Scorcher basically allows you to use, if you use R and you know how to use the tidyverse, you can build these super big machine learning models with just a few lines of like pipeable tidyverse style code and then deploy them and train them on these big systems. So you can build these really complex networks in a way that is intuitive to people who have that experience. So we're using this for lots of stuff at the Hutch. I'm just going to blaze through some examples. One is for digital karyotyping. For blood tumors, we take pictures of your DNA and look to see if there are any problems in the DNA. And so that's usually done by hand right now. There's a team of people that like pull up these pictures and look at the chromosomes and say, look, they have a mutation on this arm of this chromosome. We're trying to build models that do image analysis and make those predictions automatic, and then enable them to speed up the time to diagnosis. Another one that we're just getting started on, just starting to think about, is there's a pilot project between UW and Fred Hutch that is collecting transcriptions of the patient-physician interactions. They put a phone down and the patient and physician are talking to each other, collects all the voice data, turns it into text data. And then we want to summarize that and give the patient a summary, give the physician a summary, put the electronic health record note in the system. And so we're working on projects related to that. And then another one is early detection of pancreatic cancer, which is using sort of CT images. We extract information from those CT images and try to predict, along with the health record, what's going to happen to that patient and if they have a pancreatic cancer diagnosis or not. So this is like 3 of dozens or hundreds of applications we're hoping to enable with that Fred Hutch AI engine. So that's what we're doing here locally. Then what we're doing at national scale is we're trying to figure out how do we do this when you have multiple cancer centers involved, because most cancers are quite rare. And so like if you work in glioblastoma, there might only be a couple hundred patients that we have at the Fred Hutch, but there might be thousands nationally. So how do we do studies like that? So this is a study that's in bone marrow transplantation, and they were looking at a way to like study that across multiple different institutions. You can see lots of institutions listed here. This grant that they got to do this study was $5.5 million. It was over 4 years. They used dozens of staff to basically transcribe and collect information about patients at, I think it was 13 health centers. And our goal is, can we turn that into something that's also very fast and we can do at scale? So can we do thousands of these studies in less than a month for just the cost of computing is an idea that we had about a year ago. And that spawned something called the Cancer AI Alliance, which we're leading. And so what that is all these tech companies donated a bunch of resources and money to help these 4 cancer centers try to set up a system where we do these kind of model building activities, but they don't just happen at one cancer center. You can do it across all the cancer centers. And so that is using something called federated learning, which is a big area of growth in regulated industries like ours. It's basically a way to build a machine learning model where you never have to actually exchange the data between the cancer centers. All the cancer data stays in each cancer center, and you just be able to build models across them without ever having to exchange the data. So that is going on right now. We're in a sprint to try to show that we can do this by October 1st of this year. A lot going on. We're doing it with structured EHR data and unstructured clinical notes, and then we're going to be adding different data modalities. Both of these things, the Fred Hutch engine and this Kaya thing, are designed so that anybody who is affiliated with the Fred Hutch or a research group that's at the Fred Hutch can use those systems to do research projects. The reason why I'm talking about them is if you have collaborators at the Fred Hutch, hopefully over the next couple of years, there's going to be lots of resources that you can work with us to do all these cool projects and really cool data that everybody will have access to. So we're building these as open resources for everybody. And we're using a model of open source scaling to scale this to, if we can hit our deadline in October, the plan is to raise a bunch more money. We raised $65 million so far. We're going to try to raise $1 billion and integrate all of the cancer centers in the United States. So then you'll have an even cooler data set to do stuff with. Okay, so that's the stuff that we're doing in building these engines. Now I want to tell you what do you do with an AI engine once you have it. And I'm going to switch gears a little bit and talk about some older work that I did building a different system that let us build AI models. And I'm going to kind of start with a provocative idea, which is, what if in the future most experimental data will not be generated by laboratory experiments, but will be generated by AI models? What if we predict most of the data that we will use in statistical models in the future? Which seems kind of crazy, but is increasingly becoming the norm, that the data we use are actually generated out of a model instead of generated by people pipetting things in a lab. So I'm going to talk a little bit about the way that we built a model for this. And this is where it's going to start at some point here. It's going to start talking about company stuff, and you can decide whether you believe me or not. But so we started this by my lab for a long time had been collecting all of the publicly available data about something called gene expression. So everybody has the DNA. You have basically the same DNA in all of the cells in your body, more or less. So why is your liver cell different than your heart cell, different than your blood cell? It's because you have genes. There's about 20,000 in your body, and they get turned on and off different amounts in your different tissues. So in your blood, certain genes are turned on. In your liver, albumin is really high, but other genes are down. And when you get diseases, those same genes turn on and off. And so people have been studying this molecule, RNA, which also became famous for the mRNA vaccines that many of us took in response to COVID. People have been studying this for a long time. So we collected basically all of the data people had ever used about that. And I used to think that was just for reproducibility and stuff like that. And then I realized it was the best training set for a machine learning model for this kind of area. And so we started working on that. This recount project is all available online for free. It's still for free. Everybody uses it. So from pharma companies to academics, it's a really commonly used resource. And it's been used to do things like redefine what genes we have in our genome. Surprisingly enough, there are versions of the genes in your genome, and we still don't know what they all are, and they're still changing over time. And so very recently, we re-annotated, using that data my lab collected, they re-annotated the genome and discovered new versions of genes that we didn't know existed until just recently, which is kind of an interesting factoid that we're still versioning what genes are in our bodies. Kind of interesting. So we started a company out of this idea of can you take that big data set that we have and can you basically generate those data? Those data are generated by, I take a sample of Tyler's blood, I extract all the RNA out of the cells of blood that he has, I run a sequencing experiment, and I measure the amount of RNA in his blood sample. And our idea was, instead of doing that, can we just describe the experiment we would have done and then have the model generate the results? And so we have this huge collection of data that's growing over time that covers measurements of gene expression from various types. And we built essentially a model kind of like the models they use for things like ChatGPT to essentially take as input metadata or descriptions about experiments and spit out as output what data would have come from that experiment had you done it. And so we have lots of ways of measuring this. We have lots of tools for saying, are we figuring out which parts come from which tissues and from which diseases and from which technical components? The main thing that we look at is, does the data look real? So I'm showing you a couple of examples here. One is synthetic data, so this is data generated by our models. For that same experiment, what came out of a lab? So these correlated dots mean that each dot is a measurement of a gene. There's about 20,000 measurements here. And you can see that they're pretty correlated between the AI models and the lab, but there is some variation. But the cool thing is you compare that to, this is just two labs measuring the exact same thing, and it's basically the same kind of correlation that you would have got if you just measured two different labs, comparing a lab to an AI model, which is kind of crazy. So we've started generating huge collections of data like this. So I was part of the early days of this project called GTEx, which is a very famous gene expression study that was funded by the NIH with millions and millions of dollars. And we have been generating a free version of that we generated with our models that we made freely available for people online. So if you want to play around with it, you can go to this QR code and check out that free data that we are starting to give out so that people can compare them. And when you look at that data, it clusters. I'm showing you a couple of things here. One is a clustering diagram that shows on each of the columns, you have a sample, and on each of the rows you have, or sorry, each of the rows, each of the columns, each of the rows you have a sample, each of the columns you have a gene. And so what you're seeing is the AI and lab samples don't cluster together. They actually cluster more by tissue here. And so what that means is that our AI model is generating liver samples that look like labs' liver samples, not like AI samples. You can't really tell the difference between the AI and the lab, but you can tell the difference between the tissues. And if you generate a plot, a dimension reduction plot that has the AI samples and the lab samples overlaid, you see that they kind of land on the same PC space as each other for the AI model and the lab model. So it's kind of a cool illustration. So that's cool technology, and lots of people are now using that technology to do experiments we couldn't have ever done before. Just to give you one example, people are really interested in Ozempic right now. Everybody probably heard a lot about Ozempic, which is this drug that is probably the most widely used drug in the world at this point. And people are trying to study the post-market effects of what happens if you take Ozempic. And you can use these models to do experiments you would never be able to do before, like you can measure what happens in people's brains when they take Ozempic. And the only way you'd be able to do that study in real life is if you like cracked open people's heads and took brain tissue and then measured it. And so it's an impossible study, but you can kind of do it with these AI models. So it's kind of cool. So the question is, I've been working on that. Tyler and I have been doing other things with machine learning. He does verbal autopsy work, but there's lots of other places where this happens in the social sciences and the biomedical sciences and the physical sciences where people are making predictions and then they're using those predictions as data. And so what happens when you do that? How do we actually handle that case? And so the problem is you take some high dimensional data, you make some prediction about some piece of that data that's missing, and then you fit some kind of regression model. This is where we kind of transfer into the stats part of the talk. And you fit some kind of regression model to relate the predicted data to the not predicted data. And this problem, this sort of stylized problem, is becoming incredibly common across all of the sciences. And so the challenge is it doesn't always work great if you just treat the predicted data as if it's real data and ignore the fact that you predicted it. So I'm going to just give you an example of what the kind of problems this can cause. So imagine you fit a pretty simple model that relates gene expression to some drug dose, say Ozempic, that you care about. You can fit a regression model like this if you have both the gene expression measurement and the drug dose. But with the models that we are putting out, you get predicted gene expression instead of drug dose. So everything's a little bit off. It's not exactly right because you're not getting real gene expression data, you're getting predicted gene expression data. So what does that do? I'm going to just show you a really super simple simulation to show you an example of how that can go wrong. Imagine I have some covariate X that I simulate from a normal. I have another variable Z that I simulate from a normal as well. I'm just going to fit a regression, or I'm going to generate data from a regression model. And then I'm going to fit two different regression models. One, I'm going to fit a regression model that comes when you use the observed data, and one I'm going to do when I fit the predicted data. So if I do that and I do it over and over, I simulate that over and over and over again, and I plot the sort of data, you can see that the data for the predicted data are much narrower than the data that you observe that are the real data. So the data that come out of a model are frequently much more narrow and close to a regression line than the data that comes out of the just collecting observed normal data. And so what does that mean? It means if you do a regression model that relates y to x, you will get standard errors that are way too small. And so you get anti-conservative inference. So this is an example of that. If you do this over and over again and you estimate the variance of that coefficient, it's much smaller when you use predicted data than when you use observed data. So that's one example of the things that can go wrong. It can get way worse once you get out of linear model territory and into things like logistic regression and things like that, or survival analysis, where now I'm looking at beta coefficients estimated for a logistic regression model if I use the predicted y, and the same coefficient estimated if I use the observed y. And you can see that there are like these big discontinuities where you get really different results if you use the predicted data than if you use the observed data. And the same thing can happen if in the t-statistic space, where you just get really weird t-statistics that don't. You'd hope they'd lie on the same line if the predicted data worked just like the observed data. They don't. So we get these really weird behaviors. So one of the things that we've been thinking about is how do you deal with this particular problem? So it turns out this one, like I said, is popping up everywhere. This is an area where you take genetic data, you predict gene expression data, then you relate genetic data to gene expression data. So that's a whole field. It's called transcriptome-wide association studies. So there's a whole field that does this. And historically, the way they do it is they don't correct for the fact that they've made these predictions. They just fit regression models to the predicted data. Another one is for doing reference mapping of transcriptome data. The same kind of thing happens where you're doing gene-based associations. You have a prediction that you're making, and you're trying to associate those two things. And often people don't correct for it. Another one is when they do single cell. There's another thing called single cell RNA-seq. It's another cool measurement technology where we don't measure what's happening in a bunch of your cells. We actually look at each individual cell and measure the RNA in it. And for that, we try to predict what kinds of cells that we're looking at, whether they're white blood cells or red blood cells. And those predictions of what cell types are then used in inference models to say what genes are different between cell types. So that's another example of making a prediction and then using it in inference. Another one is in polygenic risk scores, where we predict your risk of certain diseases from your genetic profile. And then people associate those with demographic characteristics, with socioeconomic status and other things to try to see what is the relationship between disease risk and those other characteristics. And then another one is the one kind of like what happened with Suchi's problem. So this is when you're doing models for sepsis. If you actually want to look at the data for sepsis in an electronic health record, we actually don't. You don't actually see, it's not just labeled sepsis somewhere in the data set. It's actually, you have to extract from the physician's notes that they had an infection. So there's prediction models that predict whether you had sepsis or not. And so then they use those in these same kind of inferential models. So the robots aren't quite ready again to be used directly. And so how do we actually correct for this problem? And so we originally called this post-prediction inference, but have recently updated it to be called inference with predicted data after some advice from outside counsel. And so this approach is basically how do you do statistics when the data is predicted? So this is the first paper we wrote about that a little while ago. I think it might have even been pre-pandemic. So it feels like forever ago. But now this area is getting very popular. So there's a paper that came out in Science just a little while ago doing the same kind of challenge, same kind of idea with a different kind of method. And then there's a bunch of methods that are getting produced by a group out of Wisconsin. And then there's a paper written by folks here in the stat department. And then there's another paper that's written by Xiaoli Meng and a bunch of folks over there at Harvard. And so like and Zhihang Lin. And so this is sort of an area that's really getting a lot of attention right now, is like, how do you do statistics with inferred outcomes? And so we wrote a kind of cheeky review called do we really even need data, that kind of summarizes this whole area. And it's kind of a fun and easy read if you're kind of interested in the area and figuring out what's going on with it. So this is the basic problem. We have like a regression model, and we're going to replace the observed outcome with the predicted outcome. And the problem with trying to figure out how to do statistics with that is like, if the machine learning model is very easy, if you're using a linear regression model to make the prediction, then you can probably work out all the math and get an analytical solution to what the variance components look like and the bias and all that kind of stuff. But most of the predictions we get these days do not come from a simple linear model. They come from a 405 billion parameter model made by Meta. And so how do we model what's going to happen if we, A, might not know what that model is, and B, it's really complicated. So we've been thinking about ways to do that. And one of the challenges is many of the machine learning models aren't even like made, all the like components of that model aren't even made public. Originally, OpenAI was OpenAI because all of the parameters and everything about it was open. Since then, it's very, very closed. You can't get access to all of the information about how those models are built. And most of the cool new machine learning methods that are coming out, this is an example from my field about a predictive algorithm in breast cancer, where they were missing almost all of the information when they published the method. So you really can't reproduce what was happening. So you kind of have to assume you don't know how the model is working when you're doing this inference. But a trick that we observed was that once you have the some observed values of the outcome and the predicted values of the outcome, kind of regardless of what algorithm you use, very frequently, there's a simple relationship between the predicted values and the observed values. So it looks kind of the same whether you use k-nearest neighbors, support vector machines, neural networks, random forest, pick your algorithm. There's often a very simple relationship between the predicted outcomes and the observed outcomes. And you can use that to sort of try to fix up the inference in post. So we use this kind of trick where we're trying to model this relationship. And again, this is a complicated function, so we'd rather not have to figure out all the operating characteristics of it. But maybe we can figure out if we can use this really simple relationship. And when you do that, you're making a big assumption about what you're making from this step to this step. You've made a big assumption about the relationship between YP, Y, and X, that joint distribution. But often you can get away with it that you can do this kind of thing. And then you can use the YP given Y relationship that's quite simple to figure out the inference. And so the structure of these problems is quite common. You have some observed data set. You may or may not have a training set where you're building a prediction model. Sometimes this is like done in secret by a big company. Sometimes you do it yourself. There's a testing set where you're actually testing that model, and you probably have some labeled data. And then there's a validation set where you don't have any of the labeled data. You only have the predictions. So that's kind of the structure of how these problems usually look. And there's usually, and the way to think about it is there's three things going on for this kind of problem. There's some state of nature, real underlying model that relates the outcomes to the covariates. Like we don't know what it is, but it's out there. It's probably complex. There's the prediction algorithm that we're using that might be arbitrarily complex and is really designed to try to mimic what's happening in the prediction model. And then there's the inference model that's usually quite simple that we want to do, like a Y on X regression, linear regression model. And so we're going to have these three components throughout the sort of modeling. You kind of have to keep track of the bookkeeping to figure out how all of this works. So there's three data sets, the training set, or the labeled set, and the unlabeled set. And you have three pieces of data: the outcome, which is Y, the covariates that you're going to do a regression model on, that's X, and then a superset of those covariates that are those covariates plus anything else you want to use for prediction, the kitchen sink. Throw everything in there to try to make a prediction. So we have those in the training set, the labeled set, and the unlabeled set. If we have a training set and we're doing this part, we fit a prediction model in the training set that relates the outcomes, Y, to the big collection of covariates, and we make some predictions. That's fit in the training set. Then what we're going to do is we're going to try to model the relationship between the labeled Ys, the actual outcomes we have, and our predictions of those outcomes using the covariates. And we're going to use, for now, a simple linear model to relate those things. So that's that relationship, that's borrowing that relationship that we started with. And then we're going to generate new data in our unlabeled set by essentially using this relationship model and our predictions to generate new data in our unlabeled set. And then if you do that, you can fit a regression model that essentially both corrects the beta hat by using this unlabeled data to correct the bias that you get from the prediction algorithm, and it fixes up the variance because you have a component of the variance that comes from how variable is your prediction from the relationship model, and a component that comes from the usual regression variation that you get. And you can work all this out, but this is at a high level what we're doing conceptually. And so if you do this with this key observation, you can then go back and, can skip through this, through a simulation. What you can do is if you simulate a bunch of examples now, like the one I showed you at the beginning, and you do totally uncorrected data analysis where you just treat the predictions as if they were observed, you get really low standard errors. Like this is the standard errors with the predicted data, and they're much lower than the standard errors with the observed data. That's why the orange ones are down here. And if you use these corrections we have, you get corrected inference for there's a bootstrap version and an analytical version and details in the paper if you want to read about it. You get corrected inference that looks more like what you would expect, which is basically you get the same inference with the real outcomes that you got with the predicted outcomes. Cool. You can do the same thing with t-statistics, and you get the same sort of behavior where the t-statistics, the orange t-statistics are the uncorrected analysis, and you see that they deviate from the line y equals x because the statistics that are used with the predicted outcome tend to be very optimistic about the inference and give you anti-conservative results. So this happened. We wrote this paper. And then a little while later, Daniela Witten and Keshav wrote a paper where they pointed out that this doesn't work when a couple of different things happen. One of which is if you increase the sample size of just the validation set and leave the other two sample sizes the same, that approach that I just showed you starts to produce basically biased results. So the red boxes start to drop off the line here. So you start seeing more bias as the sample size gets bigger and bigger and bigger. And that was a bummer. We were not happy to see that. In our paper, we made some assumptions that were pretty strong assumptions about the sample sizes of the various sets and about the ways in which the prediction model worked. And they pointed out that these weren't working that way. And so not a great feeling when your method comes out and then somebody's like, hey, it doesn't work in all these circumstances. So we were like, uh-oh, let's see if we can fix that up. So we spent a little time fixing it up. This is not out yet, but it's coming out very soon. We're actually sending it to the journal like this week. So we did this moment-based generalization to that post-prediction inference. It gets a little more complicated, but there's two main pieces to it. So it's the same thing. We have training set, labeled set, unlabeled set, same regression models here. And then the big difference is there's two things that we're doing. One is there's this eta term, which represents the difference between the linear regression form of the y equals predicted y line. And sometimes there's a bias term that we were just ignoring. We were assuming it was zero. We were assuming the linear regression model perfectly captured the relationship in y and predicted y. Turns out that's not always true. And then the second piece is down here, and I'll talk about that in a minute, which is we extended the variance to basically be able to, with a moment-based estimator, to be able to capture what happens when you have that bias term. So this term right here basically helps us capture if there's bias that's away from that linear regression model. And then this ratio of n's right here is because the component of the relationship model that we're estimating depends on the test set size, and it shouldn't go to zero as the labeled set size goes to. If the labeled set size gets infinite, that variance in the labeled set should go to zero, but that shouldn't happen for the test set size if it stays fixed. And so you have to like input an inflation factor in there. So if you do this, you can kind of see that this is comparing a bunch of different methods, but you can kind of see for the post-BI original versus our proposed method that if you have different sample sizes, if you have the same sample sizes, both methods kind of work. If you have different sample sizes, you start to lose coverage from our original method, but the new kind of correction maintains coverage even as the sample sizes are different and when you have bias terms. And so essentially what this is doing is figuring out a way to do inference even when you don't have the observed data, you only have the predicted data. So here I'm showing you examples of p-value histograms that come from these methods. So classic is if you had observed all of the data just in the test set, so doing inference just in the test set. Naive is assuming that all of the predicted data is just data and doing inference that way. Post-BI is the correction I just talked about. And Oracle is if you knew all the real data. And so what you see is the p-value histograms are uniform when there's no effect, like you would expect for the classical approach and the Oracle approach, and a little conservative for the post-BI approach. For the naive approach, you get way too small of p-values. Same thing for coverage. You essentially get correct coverage for classical, correct coverage for oracle with much smaller confidence intervals as you'd expect, bad coverage in the naive case where you ignore the fact that you predicted stuff, and you recover the coverage that you would get from the post-PI approach, but you get slightly wider confidence observed all of the data, which is kind of what you'd expect. You kind of lose something for the fact that you predicted things. And then if you actually do have an effect, classical finds some stuff, naive finds lots of stuff, but mostly wrong. Oracle does a good job of finding stuff, and we're more conservative than if you did the Oracle approach because, again, you lose something for having made predictions. And the same thing for the coverage of the confidence intervals as before. So this approach of doing inference with predicted data is now being used in lots of different areas. We chose 2 areas for our original paper, verbal autopsy and gene expression analysis, but now people are using it for lots of other areas as well. They're using it in electronic health records. This is a paper from us, but there have been a lot of other papers in that area now. They're also using it a lot in genetic association studies. It's a really common thing. And then there's more work in verbal autopsies. And then in a paper to be submitted, we've collected like 60 papers from like 60 different areas of science where people are doing this. So it's an incredibly common thing that's starting to happen across all of science. And so I think this is a cool area. The method Tyler and I came up with our student Sarah was great, but it was just a start, and there's newer, better methods now. Our students and postdocs like Kentaro and Hoffman and Steve Salerno and Awana Fayez are working on new methods that are better in specific areas of application. But I think this is like a growth area in statistics. I think you're going to see a lot of new cool stuff. This is a fresh territory, which is always exciting if you're a grad student or a postdoc to be thinking about. So we can work with machines if we like figure it out, where we both do the machine learning and we figure out ways to deal with those machine learned data in a way that respects that they're predicted. So the stuff I talked about today, obviously I talked about a lot of stuff ranging from building these engines to what do we do with them to then how do we deal with it once they're predicted. Anything I talked about that you think is interesting, good, or right is probably from somebody on this screen, and anything stupid or wrong that I said is almost certainly due to me. And so thanks to people here in the stat department, thanks to people all over the Fred Hutch, people at the company, to our awesome Fremont Brewing Company get-togethers to talk about stats and post-prediction inference. And thank you to all of you who stuck around till this late on a Friday to listen to this talk. And if you are interested in any of that stuff I talked about, it's all on the internet, and you can go find it there. Thank you very much for your time and attention. Cool. Could you?
Robbie said, you mentioned that it's becoming more common that there are AI-generated data sets and resources. Not familiar with AI-generated data. These AI data sets explicitly mention that the data sets were developed by AI. And also, we think that these AI-generated data sets will be favored over human-generated data sets. An example, CCGA being replaced / favored over an AI-generated cancer resource.
So this is all pretty, I'll answer both of those questions. So do people label that the data is AI generated? Yes, most journals require that. Like you have to say when it's AI generated or not. And so yes, people are labeling it as AI generated or not. Though there's like a super fascinating paper that I don't know if any of you have seen, which shows the rise of the, what is it called? The emphasis, the long emphasis in writing. What's it called? The double emphasis or something? Double emph. Anyway, there's a kind of punctuation that that's it, M dash, that is very uncommon in most writing. But if you plot a trajectory of how often it's used in writing since the release of ChatGPT, it has exploded in usage. And so there are many suspicious places where it's possible that ChatGPT has been writing scientific papers. And we know this by the use of this M dash that is like exploded in specific areas. So you're supposed to label it, whether everybody's labeling it or not, maybe not. And then will one or the other be favored? I don't know. I think the thing that excites me is many of these experiments that we do, say these gene expression experiments, they're very interesting. They help us solve really hard problems. They also take a long time to do. You have to like go get samples from patients. You have to get those samples in. You have to do a bunch of biochemical work with them. You have to send them off to a sequencing center. You have to get them sequenced. They come back. You do a lot of bioinformatics work. And then you get to answer the question that you started with all the way back at the beginning. If there are ways that we can make that faster so you can answer questions faster, I think that's super interesting. And I imagine some people will adopt it. But I also suspect that some people will be skeptical for a long time as to whether that's really works or it's worth doing. And so I don't know. We'll see how fast people adopt it. ChatGPT got adopted pretty quick, it seems, but I could imagine it. And you kind of see with like AlphaFold, we had a recent UW Nobel Prize, David Baker, who just won it, the Nobel Prize, like not that long ago, won it for essentially doing this, like basically generating ways that proteins fold that are completely computer generated. They used to do that all with laboratory experiments. Now they do it almost entirely with AI, thanks to the models that David and his lab produced. So in certain areas, I think it's for sure going to be the path. Like there, it's like super widespread adoption. Lots of people are using it, lots of different reasons. So probably depends on how good the models are, you might guess.
Ross Prentice has a hand raised.
Great. Ross, do you want to just, let me see, I think, am I here? Can you ask Ross? I don't know if this will, I can't hear him. You may have to type it. Sorry, Ross, I don't know if it,
I'm getting there, Jeff. Can you hear me now? Jeff, can you hear me now? Oh, mute.
on your computer very quietly. You might have to say it and it will get repeated.
Okay. Um, I'll try. So very nice talk. Thank you.
Thank you.
And it occurs to me that a couple of points that there's quite a few analogies of the extensive work you described with the smaller scale work we're doing on dietary epidemiology methods, where we generate estimates of certain dietary intakes from metabolomic data.
Yep.
It's a place where you can, if you can hear me, I'll continue on. Can you?
Yep, we can hear you. Yep.
Okay. So at the beginning, if you just rely on self-reported dietary data, it all has some strong unreliability features, which in my mind means that it doesn't matter how large a data set you get or how much machine learning you do, you have no assurance that you're going to get usable information. And
Yep.
it's sort of like in the method you described, the validation sample is a key. In our
Yep.
case, we have a feeding study. We don't assume that we actually have the actual intakes in that feeding study, but we have them plus some random noise. And I think that's the key to a lot of the methods you were describing toward the end, that for the narrow confidence intervals and all of that, you're able to adjust for a noise component to the prediction. But if there's systematic error there, that's not going to work, I don't think. So there's a strong connection with measurement error methods in the X's,
Yep.
and which is not a very well developed field, but there might be something to learn by comparing notes on some of the efforts that we've been making in a public health area, not nearly with as extensive a databases as you have, but we have metabolomic profiles in blood and urine with 1,000 or so measurements in maybe 100,000 women. Well, we don't have all of those in 100,000 women, but in intermediate sized subsets that we then use to make predictions in cohorts of 100,000. So I'll stop there.
No, I think that is a very relevant equivalent kind of problem. And I think there are many connections between what we're doing in a few different areas of statistics, one of which is measurement area error models, where we can definitely borrow a lot of ideas and strength from understanding those models and how they're used. So Yeah, I think that it's a super related discipline. There's been a couple of decent review articles starting to come out about the relationships between what we're trying to do in measurement error models. Um, but it'd be cool to look at that data. So yeah, we should talk, Ross.
Yeah, I'd like to do that, Jeff. But what about if all the data on X's are unreliable? You're stuck, aren't you?
Am I
You
stuck?
need, you're stuck. If all of the data that are available in these extensive databases you're combining are unreliable, that is, maybe they have some systematic biases that are not really recognized or haven't been assessed to be allow correction. So it sort of relates to one of the early questions, what do we do when we have machine learned everything? I think that was a little tongue in cheek, but it
Yeah,
seems like there's a lot of topics, and public health is the area that I'm most familiar, that we really often need better data, or we have little likelihood of getting the answers that we need.
yeah, agreed.
Yeah. So even the nice papers you referred to by Catherine Henry had to go to a nicely embedded randomized trial, not really
Yeah,
generating new data, but getting data in a form that can avoid certain kinds of biases to make progress.
totally.
Yeah. Thank
Yeah,
you.
agreed. Right. There's a question in the back, but and I'm also like, if people need to go or whatever, it's 4:30 on a Friday. So I appreciate you all being here. But if you have to go, I won't be offended. Yeah, hit me.
My impression outside of the moment is that it feels like a little crowded area with lots of people like just kind of stepping on each other's toes. Like there's lots of applications all doing like the same thing, and like
Yeah,
you have all these like conflict that's people being like, look, we kind of did this with like AI killed you a super long time ago or whatever.
sure.
Weird little like infights on all of it. I don't know. I was curious if you agree with that perspective, or if you have anything to say about it, or feel like there's on the meta that field of research.
So there's only been 2 times in my career where I feel like I've done something that's worthy of other people talking about in the stats world. And like one of them was this SVA approach that was like for correcting batch effects. And like I published it when I was a grad student here. And immediately everybody started doing that same thing where like everybody was jumping all over each other and like writing a bunch of papers and all this kind of stuff. And I sort of backed away slowly after that happened. But so much interesting stuff came out of that, so much better than the thing I published originally, that like took that field forward in ways that have now had like huge impacts across the field of biomedicine. And this feels a little bit like that, where like we had kind of an idea, which I think was pretty great, and I'm happy about how it worked, but we're already seeing better methods coming out that I think than what we started with. I mean, like no discredit to us, but there's some smart people out there working on it. And so I get excited about it because it feels like a super important problem. I don't think anybody's like totally nailed how to do this yet, and especially for specific sub cases, I think people haven't, like we have people are working on it for survival analysis, and like everything we do here probably doesn't work for survival analysis in the way that we hope it will. And so there's somebody's got to figure that out. And so I think there's lots of stuff out there left to do. I do think that when that happens, when everybody notices that a problem's important, then like there tends to be a little bit more tension because everybody's trying to like, it's a little gold rushy. And so that can be a little stressful. If you don't like that kind of stress, maybe not the place to be hanging out. But at least from my perspective, I think Tyler probably feels the same way. Like I'm stoked when other people show us how they figured something out that we didn't figure out yet. Like I think that's how science works. Like I'm excited when they do better than we did. Not everyone feels that way, but that's how I feel about it. And if you kind of feel that way, then it's not stressful. You're like, yeah, oh, there's a cool thing from causal inference that does this. Awesome. Let's figure out how to do that. So I don't feel too much stress from that. Some people